Hello and welcome to Module 2 Biology, Segment 4 on DNA, protein synthesis, and muscle growth. Today we're going to go over kind of the next step. We've already discussed the cell membrane, the organelles, a little bit on the role of the nucleus, and we're going to delve into that a little bit more today and talk a little bit about genes, DNA, and a little bit into protein creation, folding, and potentially how a lot of these genes kind of interplay with a couple of examples, including one for muscle growth. The entire topic of muscle growth is kind of central to a lot of training, exercise, physiology, etc. And I think even just an overview of the complexities of it will be kind of a useful thing to see. So first, when we talk about a gene, what is a gene? Well, a gene is as it says, functional segment of DNA responsible for a protein. Now, already, as simple as that definition is, there's already a little asterisk next to it because genes can actually encode for more than one protein, as we'll see in just a moment. So how does a gene actually encode? Well, again, most of you may remember from biology that DNA is this double helix shape. It looks like a ladder that's been twisted. And on each side of the DNA strand, you have the backbone, and then you have the individual, the nucleotides that are, kind of paired up with each other along the way. Now, each one of these nucleotides in a given order, 3 nucleotides in an order, actually encode for a single amino acid. The order of those amino acids determines the final protein structure. So when DNA is ready to be converted into a protein, the first thing that happens is that a particular enzyme comes in and actually separates apart those base pairs, those nucleotides, exposing one side of the DNA. Now, RNA polymerase, as is mentioned here, matches RNA nucleotides and ends when it detects one of 3 stop signals. And these stop signals are just base pair or arrangements of nucleotides that basically say, hey, you're done. And those are pretty much universal. So what RNA polymerase does is it takes a different backbone and basically pulls it along the DNA, matching those nucleotides one by one and creating this long strand of messenger RNA, which is basically a mirror image of the DNA. So this now messenger RNA, this long RNA, isn't quite ready for protein yet. You're not quite ready to take this out and use it as kind of your blueprint. There are 2 sections of coding regions on this RNA. There are introns, which are segments that don't actively code for proteins, and there are exons. Exons are regions on this RNA strand that are actually quote-unquote valid. Like these are actually it's basically like somebody hands you a blueprint and says, you know what, there's some draft stuff here. We'll scratch this section out, we'll put some white out here. Don't look at what's under that sticky note, etc. The spliceosome is there to actually remove those non-coding segments. Now, there are so many theories as to why introns, these non-coding segments, exist. The only one you may need to know for now is that the introns that are removed, the introns that are removed aren't always the same. In fact, adjusting what is removed can subtly change the final protein. And in fact, a single gene, a single messenger RNA can be spliced different ways to create slightly different proteins. And that can be because the tissue type it's expressed in. There can be, kind of point mutations. There's even theories going that, epigenetics, which is environmental genetics, can change how certain proteins are expressed. That is a huge topic and way outside the scope of this discussion, but just something interesting to note. So now, once the messenger RNA has been created, it goes from the nucleus. Now, remember how the nucleus is surrounded by that endoplasmic reticulum, the rough endoplasmic reticulum with all the little dotted, little dots on it? Those are the ribosomes. The mRNA heads out of the nucleus and over to the endoplasmic reticulum, the rough endoplasmic reticulum. Now, those ribosomes are actually the places where protein synthesis takes place. It's where these proteins are created. So what happens is this messenger RNA is kind of transported out there, and 2 ribosome subunits, the big one and the small one, because a ribosome basically looks like a two component snowman, close around on either side of it in the membrane of the endoplasmic reticulum. Now, the RNA, the messenger RNA, starts out, it's got all these whatchamacallit, coding sections, these exposed nucleotides. A transfer RNA is a little carrier transportation unit that on one side recognizes any three nucleotides, and on the back it carries with it an amino acid. Basically, think of each one as a little car that lines up in its parking spot, and it looks for the parking spot that has the three nucleotide code that codes for the amino acid it's carrying. These transfer RNAs come along, they park on their three nucleotide, and we'll see this in the next picture. And the amino acid that they're carrying gets left there, and as each transfer RNA, transfer RNA lines up, and each amino acid gets dropped off, they connect one after the other. So you'll see these proteins, these amino acids just keep getting arranged one after the other until that stop codon is hit. Important to know, a single messenger RNA, a long strand, can actually have multiple ribosomes actually kind of working their way along it. So think of it as, just multiple builders looking at the same set of blueprints and going over it in order. It's almost like a little assembly line. So depending on the nature of the protein, you can get, dozens of ribosomes on this messenger RNA encoding for multiple copies of this protein. That is very, very critical because the process of actual the process of actual transcription, translation is not actually as fast as you might think. So being able to encode multiple proteins per messenger RNA is kind of a major mechanism. So here, the picture on the right, we can see this large ribosomal subunit and small ribosomal subunit kind of working its way along the messenger RNA. One quick thing on notation that you'll see here, that 3 and 5 refers to kind of the I don't want to say like the start and finish, but that has to do with the chemistry, the molecules that are on the end of that messenger RNA backbone. So remember, it's a backbone, it's a frame that these little nucleotides or these little coding segments are attached to. This just refers to the particular configuration of molecules at either end that scientists, that biologists use to identify basically which direction the messenger RNA is facing. Okay, so you have all these transport RNAs come in, and one by one they're organizing these amino acids. So now you have this amino acid chain, and it's a protein. So what then determines, how the protein actually does anything? Okay, well, let's talk a little bit about the structure of protein. The primary structure is just the sequence of amino acids. That's what these RNA are putting together right here. This is the particular order in which you find, alanine, arginine, glutamine, whatever the amino acid, that refers to the primary structure. In and of itself, primary structure doesn't really do much. A primary structure alone doesn't really give protein much of a function. The secondary structure, and we'll see this in the next picture, has to do a little bit with how some of these, the amino acids interact. Now, every amino acid is slightly unique. What determines protein function and structure, in many ways, has to do with the actual amino acid, not the nitrogenous portion, but the rest of the amino acid, which can be, these can be different types of ring molecules, they can be very simple small molecules, they can contain sulfur. A lot of these amino acids, each one has its own unique chemistry, which can define how the amino acids maybe even attract each other in a given protein strand. Some of them are polar, some are nonpolar. All of them have different effects on the molecules around them. And certain sequences of, let's just say of polar, polar amino acids, etc., can actually twist this primary structure into kind of a unique shape. And we'll see this in the next one. So once you have this, secondary structure, and you start to get a little bit of this kind of intrinsic shape formation, now these shapes, which can be like helixes, they can be in some cases strands with no shape, they can be sheets, which are, basically just these long strands that just kind of go back and forth and kind of pull each other into, kind of a pleated look. You'll see what this looks like in the next one and how it's represented. All of those then, those pieces start to bend and fold on themselves. You can have, you remember when we talked a little bit about the cell membrane and how you have hydrophobic portions and parts that want to get away from water? Well, if there are hydrophobic portions on this, hydrophobic amino acids on this protein chain, they will kind of fold in and cluster on each other to keep the water out. So you'll get those portions folding in on themselves, and you'll get these helixes pushing things out of the way, and you'll get this whole three-dimensional pattern that kind of resolves itself and creates the actual final protein structure. And this really determines the function. Again, we'll see that in just a moment. Last thing you have is the quaternary structure. Some proteins, some active proteins or enzymes are made up of more than one copy of the protein. One of the ones you always think about is hemoglobin. Hemoglobin is actually, if you've ever seen a diagram, it's actually 4 identical proteins. Their tertiary structure all resolves, and you get 4 of these copies that kind of mesh themselves together and to form the kind of functional structure. And the 4 of them interacting with each other actually do a lot to determine how they hold on to oxygen. Fascinatingly enough, certain things control how this quaternary structure folds in on itself. One interesting thing that affects that is blood pH can actually affect how the quaternary structure of hemoglobin resolves. And when the quaternary structure is configured one way, it doesn't hold on to oxygen as well. When it's configured the other way, it holds on to oxygen better. The body uses this phenomena and a whole bunch of pathways of this to regulate how well your body can oxygenate skeletal muscle while you're exercising. Whole fascinating segment we may get into in the future. So now back to tertiary and quaternary structure. We have this tertiary structure. We have this thing folded in on itself. This structure, and indeed the binding sites that are left open, determine function. So here we have a protein called amylase. Now, if you can see those, that orange spiral and the yellow spirals, and all those are the helixes that we talked about of the secondary structure. Those are self-creating, self-creating structures that kind of, again, everything here either lends to function directly or helps ensure that the functional region of the protein is exposed. So think about this as being something like, look at this structure here and pretend it's made of like slightly pliable plastic. If you grabbed onto this thing, you'd probably be able to, mesh it, push it around a little bit. You see how that, on the upper left, that helix looks like it's just kind of hanging there. The functional site on this protein is right underneath that, dark yellow helix, right above the light green one, kind of on the left there. That functional site is where the protein's action takes place. Nearly everything else in that protein, all those substructures, likely have to do with creating that functional site there where the magic takes place, making sure that the right amino acids have their active site. In many cases, the active site can be two exposed oxygen molecules on 2 separate amino acids, and since oxygen is is kind of a polar, its inclusion makes that particular region polar, those 2 oxygens exert its effect on whatever the enzyme does. Now the rest of the structure, even though its main role is to kind of just create that site, the rest of the structure can occasionally have binding sites of its own that can modify the active site. So, think of it as, if you push down on that light blue section there, that light blue helix, and something binds there, would that deform the active site to maybe make it able to take in, more substrate, or would it close it off or anything else? So protein structure always looks very, very complicated, but usually it comes down to 1 or 2 critical portions with the entire structure around it lending, the solidity, keeping it in configuration, and potentially opening up other ways in which other molecules can come in and modify its function. So what we're looking at here is called amylase. Amylase converts starch into sugars. Now, 2 glands in your body, your salivary glands and your pancreas, produce amylase. So what does it do? Well, remember that active site we talked about? It is a glutamate and 2 aspartates. That is the active site. That is literally what this entire protein is structured to do, is get those 3 amino acids in the entire long protein chain into the right configuration to bind to starch. That's it. So if you look at the little diagram there, The top image is a starch, which is just a chain of glucoses, chain of sugars that is held together by these bonds. What this active site does is it kind of binds onto the starch, and kind of just based on its own stereochemistry. Think about it as just like pulling these bonds apart just enough to expose that region to water, which lets it break apart. Really kind of a simple idea if you think about it. So all that amylase does is it comes in and you've got these long chains of starch, and basically they just come over and they grab on and just slightly tease and bend the starch a little bit, and water rushes in and boom, the starch snaps. That's it. That's enzyme activity right there. That's enzyme action. Now that cleaving of that hydrogen bond there that you'll see, that's called hydrolysis. So called because basically it's a function of water. So amylase is encoded by the AMY1A gene. Now, as we've discussed, a single gene encodes for a single protein. Now, the gene for salivary protein. Now, remember, every gene exists in every cell in the human body. Any cell that has our complete DNA chain has every gene in the body. So the AMY1A gene that's in our salivary glands that is activated to produce salivary amylase still exists in in our quadricep muscle cell nucleus. It's just never activated, obviously. There's no reason for it to do so whatsoever. So this gene in the salivary glands is stimulated by both sympathetic and parasympathetic systems. Now, we'll talk about this a little bit more when we talk about the nervous system, but sympathetic is, the fight, flight, or mating. And parasympathetic is rest and digest. There's so much more to it, but that's effectively what these two systems are. Each different system actually activates different genes in the salivary gland. So remember, we talked a little bit about signaling. We talked about how various systems can trigger, various transmitters, etc. And again, we'll talk about this more in the endocrine system and the nervous system. But they can trigger different messengers, different hormones, and different neurotransmitters to come in to the cell and kick off different transporters and receptors that kickstart different gene expression. So when the sympathetic system activates the salivary glands, it activates a bunch of genes that include the salivary amylase, but it actually encodes for the amount of AMY1A activated is relatively low. Now the sympathetic system, if you think about it, is all about, if you're going to go for a run, or you're going to go lift something heavy or whatever else, think about why the nervous system would want to release saliva. As it says right there, the main function is moisture for your esophagus. When you start running, if you get the salivary buildup, or you're doing any sort of high intensity physical activity, you get the salivary buildup in your mouth, that is absolutely 100% to keep your airway moist, to keep your esophagus moist, which facilitates respiration and lets you breathe. This is actually why, like I said, when you start running, you find yourself having to spit. This is all absolutely intentional, and this is your sympathetic nervous system activating genes in your salivary gland and causing, fluid production, etc. The parasympathetic system actually creates a different texture of saliva, and it does that by activating different genes, including a lot more of this AMY1A gene. Parasympathetic system is what kicks in when you smell good food, you're sitting there eating, you're relaxed, and everything else. More enzyme release happens then. Critical to know is that the reason why there's such a distinction, the reason why gene activation is kind of carefully controlled by this, is because enzymes are very costly for your body to create. You think about it, these are very long protein structures, and there's a lot of work that goes into it, so your body's only really going to produce it when it's needed. Now, what's interesting is that the prevalence of this gene, the expression of this gene, and in fact, some of these genes, people may not even have certain genes or certain copies of genes, because copies of genes can exist in multiple places in the body. Multiple places on DNA strands. Certain populations, you look at the Yakut tribe. This is a population that is located in northeast Russia that's frequently studied in a lot of population genetics. because it's a relatively isolated tribe in very harsh conditions. It's a little bit like when people study Inuit tribes and look at some of their gene expression because they eat a diet that's extraordinarily high in fat, low in starches, etc. Yakut tribe is similar. They have very, very low expression of the AMY1A gene. In fact, many people lack that entirely, whereas populations in Europe and Japan have large amounts of this gene. And if you look at the number of copies, the incidence of a gene being missing is next to none in these populations. And at some point we will talk a little bit more about how gene expression varies across populations, because expression of genes and incidence of genes and copies of genes in different populations is something that's increasingly looked at when we look at population propensity for different types of performance. For example, we look at certain populations and you say, well, why do all these great strength athletes come out of Poland, and why are all these fantastic runners coming out of this one tribe in Kenya? And certainly a lot of it has to do with with basic physiology and structure and everything else. But there's always the possibility, and it's very true. It's not just a possibility; it's a truism that the presence of different genes, some of which may be responsible for certain levels of sympathetic arousal, muscle building, mitochondrial proliferation, AMP kinase activation, et cetera, et cetera. All of these things may vary tremendously across the human population. Anyway, going back to AMY1A, when you have individuals that secrete a lot of salivary amylase and have a high incidence of this gene expression, oral digestion actually becomes very, very crucial for these folks. And in fact, proper chewing causes blood sugars to rise more quickly, causes blood sugar to rise more quickly than not chewing quickly, if you just say swallow entire boluses of food. This can be very important in things like inflammatory bowel disease, because remember, the only other site where amylase, which breaks down starches, is released is by the pancreas, which is a little further down in the digestive system. So if you are an individual with a potential irritable bowel issues or anything else, you may find that starchy foods are difficult to digest. Sometimes the simple act of chewing these foods lets this salivary amylase be produced and be created in your mouth. And it'll have this effect on starches and actually have a lot of these starches broken down by the time it even gets to your stomach. So, we're going to talk about a little bit next is muscle growth and how a lot of gene expression goes into muscle growth. Now, we can go back to modules 1.3 and 1.4 to talk a little bit more about actin and myosin. Important to note is that, again, like anything else, actin and myosin are typically thought of as muscle contraction proteins. But actin, these two proteins are found in so many cell types. The idea of two proteins that slide back and forth across each other and actually do productive mechanical work has major functions in a lot of different cell types. Any type of cell that requires any level of motility. You look at villi in your intestines and how the villi actually move food along and move things along. Many of those are much of that motion is actually controlled by actin and myosin as well. In fact, actin has a major role in things like certain cell division, cell replication, et cetera, et cetera. So without further ado, muscle growth itself, and we're not going to necessarily say this is the answer to muscle growth here, but I want to talk a little bit about what a complex process this is and how much gene expression is going on. Well, when people talk about muscle growth, they talk about the mTOR pathway. Well, what is mTOR? mTOR is called the mammalian target of rapamycin, which on its surface has absolutely nothing whatsoever to do with muscle. Rapamycin is an antifungal. It was discovered in 1972, as you can see. Now, rapamycin has a certain antifungal property, and if you look at it already, if you start to think about it, if mTOR leads to muscle growth, we'll see that mTOR is a particular receptor complex that has a bunch of downstream effects on gene expression. Now, if just gene expression is responsible for growth, you can already see how potentially inhibiting mTOR in certain ways could potentially reduce cell growth, which may be useful as an antifungal or antibacterial or anything else, as long as we can make sure that it doesn't happen humans. All right, so what causes this mTOR pathway to activate? Because the theory, one of the prevailing theories is when you activate mTOR, activating this this receptor complex is needed to actually cause muscle growth. So the gold standard becomes, how do we activate mTOR? Well, what's interesting is that mTOR activation increasingly appears to have very little to do with things like inflammation itself or any of these other things traditionally thought as leading to muscle growth. One of the biggest activators of mTOR appears to be what are called mechanoreceptors, and these are receptors in muscle cell that basically detect excessive stretch or stress. And as you can see, stretch loading increases certain metabolites compounds in the Z-band. Remember the diagrams of muscle structure, how actin and myosin overlay each other, and there are the M-bands and the Z-bands where the actin and myosin attach? Well, if a muscle is under stress, these contractile proteins are kind of getting pulled apart a little bit. And it could very well be, when they are pulled apart, the metabolites that are created in the kind of rush to repair these things that trigger the cascade for muscle growth, which would be a very, very interesting idea, because it is technically muscle damage, but it's not gross muscle damage, big muscle damage. It's simply the act of stretching or pulling on a muscle that triggers this entire growth cascade. So in this diagram, you can kind of see, now notice where it says muscle spindles in that box. These are receptors located all around muscle fibers that actually detect stretch and stress. You can see the way they're arranged. When they are pulled, when they detect this stretch and stress, they send a signal back, in many cases, back to the brain, back to the nervous system. Muscle spindles, by the way, are also the fibers that cause what's known as the stretch reflex. When you pull very quickly on a muscle and the muscle tenses, or you bounce at the bottom of a squat, that's all the stretch reflex. That's caused when the muscle detects it to be lengthened and it contracts to actually stop the lengthening. The Golgi tendon organ is the one that's located at the bottom there, and that's the one responsible for relaxing a muscle when it's under excessive stretch. When you actually do static stretching, what you're doing is desensitizing, and you're activating the Golgi tendon organ while desensitizing the spindles to let the muscle relax and achieve its full length. We'll talk a little bit about cramp theory as well and how that has to do with an interplay between these two systems. Anyway, so both of these are technically mechanoreceptors. The muscle spindles are the mechanoreceptors here that are being theorized to actually stimulate muscle growth, which means, again, the simple act of stretching and putting stress on a muscle may be sufficient to cause growth. Now, this kind of flies in the face of the idea of needing muscle soreness, of needing localized inflammation, but as we'll see, it's much more complicated than that. Because if the goal is to activate mTOR, well, let's see, how does mTOR function? Yeah, glad you asked. If this looks all but inscrutable, that is absolutely intentional. You can see mTOR1, located there in the middle, and you can see all the different systems that activate it and are activated by it. That is why this is so complicated. The muscle growth outcome is just one small portion of the arrows leading off of this complex. And we actually have a couple papers discussing, what prevailing theories on mTOR1, mTOR. What is, I think, most important to note here is that this is why anything, simply saying that, activating mTOR should be responsible for muscle growth. Well, look at how many things activate mTOR. Look at how many things mTOR activates. And think about the fact that many of the things it activates may also be activated by other compounds or by other metabolites, because if mTOR triggers something downstream, it's entirely possible that something else may trigger something else downstream as well. So when we're talking about any sort of absolutist, what I call kind of a reductionist method of discussing muscle growth or any complex system, this diagram precisely explains why. When someone says, oh, well, AMP kinase inhibits mTOR, well, yes, but so do half a dozen other things. And half a dozen other things activate mTOR. And, there's AMP kinase, and the reason why I mention that is because AMP kinase is, as we discussed before, it's caused by localized hypoxia in cells, which is, usually a product of endurance training. And they say, well, if you activate AMP kinase, you shut down mTOR, and therefore, if you do endurance training, you temporarily shut down the body's ability to create muscle. Looking at this diagram, this is why you see something like that and you say, well, can we really make that claim just yet? So the answer is probably not. And if nothing else, like I said, this gives an interesting idea of why the research on all of this is so complicated. And like I said, we'll be able to check out some papers on it. All right, well, so here we go. It's activated by so many things: nutrients, growth factors, DNA damage, stressors. It's responsible for cell growth, motility, and inhibits autophagy. All these things associated with growth. Now, notice here, remember our discussion on autophagy and how a reduction or inhibition of autophagy may be responsible for aging? Well, they are finding that reducing mTOR activity can prolong life. Again, AMP kinase, momentary inhibitor until energy balance is restored. Item 5 here discusses why we can't make the simple claim that inhibiting mTOR inhibits muscle growth, because there are downstream components that you can activate without it. And also, activating it doesn't always lead to increased MPS, which is muscle protein synthesis. So again, just looping back to the idea that activation of a single receptor. Now, this mTOR, everything you saw leading off it was theoretically a different gene that mTOR activates. And remember, gene expression is very much dependent on stressor. Well, more than one thing can activate a certain gene. And mTOR may activate a lot of genes that eventually encode for contractile proteins, et cetera, but obviously it can't be the only thing that encodes for these things. Because every cell, like I said, in the body has a copy of every gene. And every cell in the body, we said actin is located in almost every cell in the body. Well, if mechanical tension was the only thing that activated mTOR, and mTOR is the only thing that encoded for contractile proteins like actin, and we said actin is needed in other cells for things like, creation of microvilli and for, cell replication or division, then how on earth would we activate it every time a cell wanted to divide? We simply couldn't. So none of these statements can be absolute. And so what all of this comes back to is, if you want to make gains, just lift heavy weights and don't worry about no, I'm just kidding. No, we definitely want to delve into a little bit more. But again, snapshot of complexity. So like I said, it appears to be a master regulator of anabolism. It may activate and potentiate the expression of numerous enzymes and proteins that are responsible for growth in various anabolic processes. It may also inhibit the expression of certain proteins responsible for catabolic processes. Remember what we said, autophagy requires, in some cases, like protein tags? Well, let's talk about this for a moment. When a skeletal muscle is damaged or undergoes stress, the actin, for example, may undergo a certain amount of damage. And actin, once a protein structure starts to break, it's not like things could just come in there and repair it and throw amino acids back in. That entire protein structure may, in fact, lose its integrity. Now, remember we talked a little bit about chaperone-mediated, autophagy. What happens is that there is actually a specific protein that is generated when muscles are damaged, which comes in. Now, when muscles are damaged, a certain growth factor, so a pro-inflammatory factor like leukotriene. People talk about prostaglandins. The prostaglandins that cause swelling and soreness and everything else. Those actually may go to the cell and go to the nucleus and encode for specific protein tags whose one job is to find damaged actin and tag it for destruction. So think about that for a moment. You cannot repair a muscle that has multiple damaged actin strands. You'll just be layering on more and more strands, and the old ones will be broken and nothing's going to work right. That's very bad for a cell. So to repair a muscle that has been damaged, to repair some of that damaged actin, you need quote-unquote, catabolic pro-inflammatory signaling systems to go in there and to carry out gene expression for these specific tag proteins that come out, attach to the broken parts, and carry them to the lysosomes to be destroyed. Then the cell can take the amino acids from the destroyed one and make new intact actin filaments. So you can see, again, not a simple process, a lot that goes into it, but all kind of somehow, hopefully now, is kind of starting to come together. You can see that, okay, this is, if gene expression is the goal here, and there are different things to trigger it, and everything has a purpose, even catabolic processes have a purpose, et cetera, et cetera. So there you go. Now, another interesting thing. Remember I said that this muscle damage can induce autophagy? Well, when people say, "Oh, well, you need to exercise to stimulate this catabolic process," remember that potentially the type of self cleaning that goes on when a muscle is damaged is simply cleaning up damaged muscle components. It may not be cleaning up other damaged organelles. So critical to remember that every single stressor may release multiple different signaling pathways, which activate multiple signaling pathways, which activate multiple genes, which carry out a whole range of activities. So, looping back to muscle growth, tension is growth. It may not be related to notable metabolites outside the muscle, like lactate or anything else. It may have to do just with metabolites within the sarcomere, within the contractile portion itself, which may activate those muscle spindles, those mechanoreceptors, basically let those mechanoreceptors know what's going on. Now, repair metabolites are potentially unrelated. So anything we talked a little bit about those prostaglandins, they may not be related to muscle growth directly. In other words, their presence doesn't grow muscle, but their presence may still be required for muscle growth. Looping back to that for just a moment, a lot of the discussion now about muscle growth has to do with how mechanical tension is sufficient, and metabolite training and all that doesn't really make a difference. And what some people have asked is, well, when we talk about anti-inflammatory compounds potentially reducing the potential for muscle growth, well, if inflammation isn't necessary for muscle growth, if it doesn't stimulate muscle growth, then will anti-inflammatories hinder muscle growth? Sounds like not. Well, not so fast. Anti-inflammatories aren't needed to stimulate muscle growth, but since those anti-inflammatories, inflammation isn't needed to stimulate muscle growth. But if you blunt that inflammation, you blunt the amount of prostaglandins getting to the muscle. The prostaglandins are responsible for cleaning up the damaged tissue, which allows the muscle to grow bigger. So while more metabolites and more inflammation doesn't mean more muscle growth, you cannot get muscle growth without them, because the factors that are needed to stimulate muscle growth will also require, later on, these pro-inflammatory compounds to clean up. Does that make sense? Because remember, if we stretch a muscle, it's not saying the more damage, the better. Because that's not the case. It's not like if you stretch and beat the heck out of a muscle, it's going to grow back bigger and stronger than if you didn't beat the heck out of it. All you have to do is induce enough stress to produce a small amount of stress on the muscle spindles. You're still going to elicit a little bit of damage. And you cannot repair and regrow the muscle without a sufficient stimulus to cause some damage. The more damage does not mean it's better, but that damage still exists, and you still need pro-inflammatory compounds to come in and clean up what little bit of damage there is. If you blunt that response, that repair response, you may very well reduce the body's ability to generate new contractile proteins. Worth reviewing in your book for this segment, pages 446 to 453 in OpenStack's Anatomy and Physiology. Worth going back a little bit and looking at muscle contraction and reviewing segments on cellular respiration.